Ladies and gentlemen, welcome to Gresham College. My task this evening is to talk to you about the future developments that are going to happen in medicine, particularly in eye care. Some of the things that I will be talking to you about are already in the clinics. Many of them are in translational research, and only some of those will actually become a reality. In fact, many of the things that we see in the clinic will possibly die out as other treatments are shown to be more effective in the future. It's a very exciting area. I'm going to actually begin, though, with the old times, and we're going to move forward to explain how gene therapy came about. Now, originally, we had problems with anti-infectious agents because until the dawn of antibiotics, there was really only ways of killing bacteria that could also damage the person or the animal you were treating as well. But as soon as antibiotics were developed, antibiotic resistance became a problem, and became a problem very quickly, and spread very quickly through mobile genetic elements. And there's a number of different ways that this could occur. These were studied in detail in the 1950s and 60s and set the scene to allow scientists to think about using mobile genetic elements to replace defective genes in humans and animals. This isn't all the story, and there have been many problems and pitfalls on the way. It's rather more difficult to do than you will get the impression from the simplified version I'm going to give you tonight. But nevertheless, for previously untreatable eye diseases that would inevitably lead to blindness, there is now hope and some very encouraging results coming from clinical trials. And then finally, I will very briefly mention some of the other developments that are going on with prosthetics, optogenetics, and stem cell transplants. The story begins with Gregor Mendel, who was a monk in this institution. He was discouraged from doing his experiments on hybridization with mice by his bishop. He commented wryly that the bishop didn't understand that plants also have sex, and went on to study the variations in the pea plant. Luckily for him, he chose 7 variations that occur independently of each other. But he worked very hard and made thousands and thousands of generations of these peas. And what he discovered was something that was very, very important. What he discovered was that the primary parent generation, if they were purebred, led to the first generation, the F1, having just one characteristic of either the mother or the father. But the next generation, from self-pollination, the recessive feature reappeared. And this reappeared consistently in a 3-to-1 ratio. And the numbers he had were really large. So we had 700-odd purple flowers, 224 white flowers, and this ratio is almost exactly 3-to-1. These are large numbers. They were criticized by Fisher in the 1930s and 40s, but subsequently it's been found that, taking into account how the experiments were done, they represent true results, and that Mendel did not actually invent his findings. So he tried to find an applicable mathematical law. He trained in mathematics before he came to this part of the world to do these studies in the gardens. And he came up with the ratio that you can see here, capital A plus A little a equals A plus 2 big A little a minus A. Now, some of you with horrible memories of O-level maths will recognize that as a quadratic equation. What he had found was a fundamental mathematical law of God, which was one of the things that had actually driven religious people in studying science for over 1,500 years. Furthermore, it didn't just apply to the flower colour. It applied to the seed colour. it applied to the colour of the pods. And every single time he did these experiments with large numbers, he was finding that the second generation, the recessive phenotype reappeared at a 3-to-1 ratio. Later, this was going to be interpreted by Correns and others as having two components were being inherited for each of the phenotypes. And one of these was a dominant and one of them was a recessive. This is not what Mendel did. Mendel did an extraordinary work and presented through his co-workers very good results on hybridization, which was his field. He was not ignored. In fact, his work was presented in London and was very well received on hybridization. Now, do remember that Mendel had no concept of what a gene was. By the way, nor did Darwin. He was working on the possibility of there being a gemule, but he didn't really have any concept of what genetics was about, because that hadn't been invented yet. Now, when Correns actually made the postulation that, in fact, there were units of hereditary being passed on through the generations, the next question was what were these units? The commonest thing to believe in those days was that, in fact, they were going to be proteins, because proteins had been recently studied and analysed, and it seemed to be a very reasonable assumption. And also the site of the location. Well, Robert Brown, who'd sailed with Flinders to Australia, and remember this is one of those many anniversaries that comes up, I think, next year, was the first person to document the nucleus in cells, actually in plant cells. Misha discovered a very important thing. He discovered the nuclein, which was staining in white blood cells, and that's what Walter Fleming put the aniline dyes into to make these beautiful drawings here, which demonstrate these wispy, dark staining structures within the nucleus of the cell. And many other experiments were done which showed that chromosomes were needed to pass on the information. Sir Archibald Garrod discovered that alkaptonuria obeyed a recessive trait. This was the first metabolic disease that had had its genetics worked out. And we're moving now very quickly from peas and pea colour to animals and real diseases. And this is an extraordinary moment in the history of science. William Bateson, here's the cartoon of him holding his ducks. He created the term genetics, meaning to give birth. The word gen had been earlier used by the Danish with a similar origin. And then eventually genes were shown to be on chromosomes, and then a genetic map of the chromosome was made. Then DNA was found in chromosomes in the 1930s, and the RNA, which is the ribose nucleic acid, wasn't in the nucleus. That was found to be in the cytoplasm. And then a number of other experiments went on, which were going to lead to the important discoveries enabling us to consider gene therapy. So we know that the genetic material is DNA, deoxyribose nucleic acid, and the genes are located in the DNA, which is tightly wrapped up, as you can see from this top image, to form the beautiful chromosome structures that Walter Fleming had discovered with his aniline stains in the previous century. Then Watson and Crick, using Rosalind Franklin's data, and this is her X-ray of the DNA crystallized molecule, worked out that this represented a helix. And after some experimentation, and this is their original model made out of test tube clamps and pieces of glass, and a reconstruction of this can be seen in the Science Museum today, they came to the astonishing conclusion that it was not just a helix, it was a double helix, an intertwined helix. And this is the letter to his son, which he's showing here with his excitement at what had been discovered. Now, how much is present in a gene? Well, a human genome has about 3 billion of these base pairs. Bacteria such as E. coli has less, would have 4.6 million. The interesting thing is that of all these 3 billion base pairs, more than 99% of them are identical between everybody in this room and between everybody throughout human existence. But I look in this room and I see tremendous variation in phenotype, and that means what people look like. I see variation in hair colour, mainly further towards the back than the front, I admit, and colour of eyes. Now, this 1% of the differences is responsible for all of this, and some areas of the genome are variable. So we can have alternative variations of the gene. If they occur reasonably frequently, these alleles are called polymorphisms, and they've got to occur in 1% of the population to become a polymorphism, and that would, for example, there are very few simple Mendelian traits when we look at human physiognomy. For example, the eye colour is related to many genes, but for simplicity we could say blue and brown eyes, for example. And the difference between the blue and the brown has a genetic basis. And the differences between those are allelic. So you can have people who have brown alleles and you can have people who have blue alleles. And in fact, the mutation that led to blue eyes occurred relatively recently in human history in the time after the Ice Age and was a sexual selection so that blue-eyed males would select blue-eyed females to ensure that their children were their own, because if they ended up with a brown-eyed one, they could be pretty sure that they were not the parent of that child. And that is believed why this rare allele that otherwise has no use whatsoever was so highly selected and becomes more and more common as we spread throughout Europe going towards the north and west. And it's the inability to make a protein. It's a recessive gene. The most famous example, which was in our biology textbooks, and then we went to medical school and told, oh no, that was all wrong, and now I've just been reading up some stuff for this lecture and found actually it is all right again, is the one of the moth. Now, the natural state of this moth is the peppered state, the peppered moth, and it's very good at camouflage. It's very difficult, even for those of us without macular degeneration in this audience, to actually see that moth on the normal tree. But in the Industrial Revolution, trees did not look like that. Trees looked black and brown, and so did buildings covered with soot. So you would show up very, very easily if you were a white moth, and you would become food, bird food, and that is what happened. So there was a rare allele in the moth population that allowed them to make melanin. Those that made the melanin were much more difficult to spot on the darker trees, and therefore lived to survive and to breed. And in fact, by the 1890s, the melanotic moth was the commonest form of the peppered moth in Manchester. And this is a sort of Darwinism in action. How is this code read? Well, we know that there's a pair of bases, sugar bases, which are holding the DNA coil together. And these can only pair in a certain way. What happens is the DNA is read by an enzyme that makes a message called messenger RNA, which is an exact copy. That copy, after some processing, which we will come on to, is taken up to the ribosome, which is a structure made out of proteins and RNA, and then it's translated and made into a peptide. And how this is done is very strictly controlled, and it only occurs in a certain direction. So the main things are the copy, then that's the transcription, and then the translation of that into a protein. Before the mRNA leaves the nucleus, it is spliced, as we can see here. That was exciting. We'll try this one. It's spliced, so the long copy, this bit, which used to be called junk or rubbish DNA, is taken out, and the main bits of the gene are spliced together. Now, it turns out this is not exactly junk, and some of this is going to be useful for gene regulation. And also what's important is, depending how you splice it, you can make from one area of DNA, one gene, several different proteins. And this is what's done, for example, with antibody molecules or the receptors on immune T cells. So it's a very important way of generating diversity from a limited number of genes. And it's more important than has previously been recognized. And the spliceosome is the protein complex that actually deals with doing this. I'm going to give you a little primer of bacterial biochemistry because you know what's coming next, which is antibiotics. So we need to understand why bacteria are different. They don't have paired chromosomes. They've just got one, a great big circular bunch. All their DNA is here. They synthesize de novo from para-aminobenzoic acid the dihydrofolate, tetrahydrofolate, which is going to be the precursors to actually build this double helix. They use a slightly different mechanism here of the ribosome with a 50S and a 30S. It's a slightly different structure. But again, very similarly to higher animals and humans, the transport RNA carrying the amino acid comes in, is locked in, and then moves down the chain, and eventually the protein is formed. And if it's released, this could be a toxin, such as a cholera toxin, or it might be a protein that's needed for the cell, such as the transpeptidase that makes the wall. How the amino acids are added on is step by step, so it grows. So here's the nascent chain. This is the code. This is the complement that's carried on the transport for the amino acid, and in it locks. So the next amino acid that's going to go on here is going to be red. So here we go. The next one that comes in is carrying blue, which will be locked in here, and then the pale one will be locked in here. And gradually this chain grows and grows and grows, adding on all the different amino acids that there are for making a protein. So in broad terms, there's nothing particularly different from eukaryotic cells, that's cells with nucleuses in the general terms, but in the detail there's an awful lot of difference. And it's that detail that enables us to use molecules that can kill these critters and not kill us when we use them. So in summary, before we move on to the antibiotics, we've understood now that genetics is the study of hereditary. There are discrete bits of hereditary called genes. The expression of these genes is what gives us the phenotype that makes us a black peppered moth or a peppered pepper moth. How those phenotypes come to populate is determined by natural selection, the same as perhaps blue eyes in Northern Europeans. When we have variations in genes that don't just change the phenotype in a non-dangerous way, such as brown or blue eyes, but in a dangerous way that can cause a disease, such as an inborn error of metabolism, that's very serious. And that can lead to blindness, if it's affecting the eye, or death, if it affects other parts of the body. And correcting those genes was the big challenge. It was considered, not so many years ago, to be an impossible dream. And in fact, after some accidents, the whole project nearly came to a grinding halt. I'm just going to take you back very briefly to a pre-antibiotic world, when people routinely died when they caught tuberculosis, where a cricketer, famous cricketer who'd scored many runs in Melbourne on the Ashes tour, could slip on a dance floor and die from septicemia from a grazed elbow, where the son of the President of the United States could get a trivial injury playing tennis and get it infected and be dead within 3 days. There was nothing anybody could do about this. I want you to consider the future, where if we end up with multiple resistant bacteria for which there are no antibiotics, we're back in this world again. That future is with us now and has already happened. And I'll go through and I'll show you what I mean by that. And furthermore, unless we do something about this, it won't be infrequent, frightening events. It will become the norm. And there are very few people alive in this room now who can remember when epidemics of infectious disease would take out whole communities. Dozens of children would suddenly not appear back at school. Affected people such as soldiers. In the First World War, I mentioned in my last lecture that in fact the second commonest cause of soldiers not appearing for duty was venereal disease, mainly gonorrhea. And the British Army, of course, in its traditional way, said these Christian soldiers can't possibly get venereal disease, so they did nothing about it. But much higher incidence in the New Zealand troops led to something had to be done about it. Now, there were no antibiotics. The only thing you could do was to make licensed brothels, such as Maxime's Bar, where soldiers displaying obvious infectivity and the girls working in those bars displaying obvious infectivity were barred. And this reduced the rate very much, and this was the German mechanism of dealing with it, and that's why they had a very low rate of venereal disease. And then this person, Etty Rout, whose name is forbidden to be mentioned in New Zealand on pain of a £100 fine. She's actually a hero. She was given honours by the French after the First World War for setting up these bars and providing her prophylactic kit. Between the First and the Second World War, experimenting on bacteria, we'd realised that certain chemicals could dye bacteria. Now, if they could dye bacteria, as Christian Graham had shown, could they kill bacteria? And Donach, who'd fought at Ypres, found this Prontosil. Now, the great thing about Prontosil was that it did kill bacteria, but it dyed the patient red. Now, he accepted the Nobel Prize, but was arrested by the Gestapo briefly because Karl von Ossietzky here, who had been the editor of the anti-fascist Die Weltbühne, was currently in a concentration camp for revealing the German rearmament to the world, and he had been given a Nobel Prize. So the Nazi Party were not pleased about Nobel Prizes and forbade anybody to accept one. Unfortunately, he was to die in the concentration camp of tuberculosis. Prontosil was actually not a unique drug. It had been discovered previously as a dye, so unfortunately they couldn't patent it and make a fortune. But very quickly, May and Baker over here in England did, and they made sulfanilamide, which did not dye the skin red and was far more tolerable. And a lot of Me Too drugs came onto the market, some of them with tragic effects when 100 children died of ethylene glycol poisoning with the Xylir of sulfanilamide in 1937. But these drugs changed the world. Suddenly, women did not die in childhood of puerperal sepsis. Children with sore throats did not die. People with infected wounds no longer died. This was a major advance, and it was the first of the modern antibiotics. In fact, interestingly, this German invention saved Churchill's life. When he was coming back from the Tehran conference, he fell ill with pneumonia in North Africa. He was given sulfapyridine, May and Baker, which has now been licensed to the Americans and was available in vast quantities, but was put on a strict diet that allowed him only to drink weak whiskey and soda and not to smoke at all. No cigars. He complained bitterly to his nurse. He said, "Pray remember that a man cannot live by M&B alone," which is the May and Baker sulfapyridine. The myth that he was treated with the good old British penicillin is, of course, a myth. It's also a myth that penicillin was used in these situations. It wasn't. What was used was crystalline sulfanilamide and sulfadiazine tablets. Penicillin was far too precious to be used for war wounds. It was needed to get 11,000 soldiers a week back into the fighting line who were getting gonorrhea in Alexandria and various other places. And the treatment, the old treatment, was taking several weeks to a month, and for syphilis could take 6 months. So penicillin was reserved for treating those people. We know that Alexander Fleming discovered it when he noticed that a mould inhibited the growth of bacteria in a Petri dish. His wasn't as neat as this, and in fact that's a photograph, famous photograph of that original Petri dish in the laboratory in Praed Street. Now, Ernst Chain was important because this penicillin mold, there's not enough penicillin here to treat a mouse for a day. You need to make tons of this stuff. So they started manufacturing in biscuit tins and collecting all the mould juice. And eventually they had enough. And a policeman who'd cut himself on a rose thorn in his garden was dying and had already had half his face removed surgically and his eye to try and control the rip-roaring streptococcal infection. He was given all the penicillin that was available and came out of his coma and was able to meet his family, who came to see him. The next day, they collected his urine, repurified the penicillin, put it back into him. And he survived another day. The third day, there was not enough penicillin left, and he died. So, this lesson taught the antibiotic producers that they needed mass production. So where do you go for mass production? The home of mass production, America. So they went off to America and went to the Department of Agriculture, where they were used to manufacturing vast amounts of fermentation products. Unfortunately, things went wrong. Initially, they went right. They found that if they used a corn steep liquor, it improved the amount of penicillin produced by the cultures. They also found that there were other varieties of penicillin. Ironically enough, one in a Peoria fruit market just down the road, when they scraped it off a melon, produced 10 times the amount of penicillin than the old St. Mary's culture did. So a lot of work was done, a lot of patents filed, which was what led to some bad taste between the English researchers who'd given their work for free. And then penicillin eventually comes onto the market. In 1943, the first shipment arrives in North Africa, and a big fight. Now, Colonel Edward D. Churchill, no relation recently, but maybe distantly, said that this is going to be used for treating war wounds. It was considered by Chain and various other peoples an anathema to use this for what it needed to be used for, which was gonorrhea. So the chief surgical consultant, the theatre surgeon, applies directly to Churchill, and Churchill makes the famous order that it can be used to its best military advantage, and if that means those wounded in brothels, so be it. So that's why penicillin was used to treat this disease. Now, it wasn't all just the feminists say this was terrible, everyone's blaming the women for this disease. Well, look at this poster. Syphilis, all these men have it. Women, stay away from dance halls. Now, eventually there was enough production that did enable them to start to use penicillin for battle injuries later on in 1944. And by that time, they'd reduced VD by 30-fold. It was an amazing advance. But there was still 600 incapacitated every single day. And when you've got a shortage of military personnel, this is a significant effect that can make the difference between winning battles and losing battles. So this is why penicillin was very, very important and probably made a big difference in that North African campaign, but not from treating battlefield injuries. Now, of course, we didn't invent antibiotics. They were invented by microorganisms 3.5 billion years ago for a number of different reasons, to get a competitive advantage in the hot, sweltery, slippery pool that you were evolving in. And very quickly, as soon as higher animals developed, such as leafcutter ants, this is an extraordinary picture. This isn't a lot of leaves on the floor. This is a lot of leafcutter ants carrying the leaves back to their den. And in fact, they are the major grazers of the neotropics, the leafcutter ants. What's interesting is they garden the leaves and they grow a fungus on it. Now, the problem with the fungus is the fungus has a parasite that can kill it. So what they do is they make the another culture with actinomycetes that produces many antibiotics, and this stops the bad microorganisms getting in and destroying their beautiful garden and fermenting leaf crop that they need for survival. It's a very ancient system, and in fact, when the queen flies on her flight to set up a new den, she will take a bit of the fungus with her, and she'll also take a bit of the bacteria to produce the antibiotic to ensure that fungus will survive. So when you're busily there spraying your tomato plants and so on, thinking, hey, this is really good, isn't it wonderful how advanced we are, you realise that 50 million years ago, leafcutter ants had exactly the same mechanism of looking after their gardens. Very, very quickly, people realised that if one mould, penicillium, can produce an antibiotic, there must be some others. So in Waxman's laboratory, which is an agricultural research unit, they went on a search. And they searched everywhere. They went into the soil outside. They went and scraped under fingernails. They went and had a look under stones. They sent missionaries off abroad who sent back funguses. And actually, from one of those in 1953, vancomycin was eventually cultured. And this is one of the drugs that we use for multiple resistant staphylococci to this day. The most important drug was streptomycin, because this changed the world because it allowed the first antibiotic treatment for TB. There was a great fight because Schatz, who'd done a lot of the groundwork and in fact had actually isolated some of the original antibiotics, was not mentioned in the Nobel Prize award, and he was furious. And secondly, Waxman had done some patents, and he hadn't used the money he was getting for the patents entirely for noble purposes. And a stinking fight fell out. Waxman said it's because he was jealous. He said it's because he's a crook. Eventually, the university did their own research and realized the major contribution that Albert Schatz actually made, although he did die a very unhappy and embittered but brilliant scientist who we have to recognize. Now, antibiotics are not easy to find. You have to look at a lot. So if you take 10,000 Actinomycetes species, okay, Actinomycetes, that's the soil bacteria, and you screen them, 2,500 produce antibiotics. 1 in 1 million will produce erythromycin, which is a common penicillin variant for those allergic to penicillin. 1 in 10 million will produce daptomycin. So we've discovered all the easy antibiotics already. Look how difficult it is to find a novel antibiotic now. This is one of the problems we have, because as we generate resistance, it's more difficult to find the antibiotics. Now, the mechanism of antibiotics is various. We can inhibit metabolism. This is how the sulfur drugs work, because they look very similar to PABA, don't they? So they interfere with this enzyme, and you don't make so much DNA. So the bacteria can't divide, and it dies. We can also inhibit the cell wall, and this is how penicillins work, how vancomycin works. And it sits into a lovely slot here, and it stops the cell wall being formed. This, as the bacteria grows, the cell wall bursts, and the bacteria dies. We can actually stop nucleic acid synthesis with a number of different things to do this. We can use rifampicin, quinolones. We can stop the cell membrane with polymyxins, and more recently with colistin. And then we can inhibit the protein synthesis more directly with chloramphenicol, which you'll see as an eye drop over the counter, clindamycin, tetracyclines, and a bunch of aminoglycosides that are used in hospitals but do carry with them some notable toxicity, including that of deafness. And this is how they work. They interfere. They get into this machinery, rather like sticking a glove into the wheels as they're turning round, and it grinds to a halt. But look what happened. Even in 1940, Ernest Chain had reported on strains of bacteria that were penicillin resistant. And this is before penicillin had been released as an antibiotic. So already bacteria had already worked out this biological warfare between each other. You make an antibiotic, I'm going to make something that's going to stop that working on me. I'm going to make another antibiotic, I'm going to make something else. And this had been going on for billions of years as life crawled up the greasy slope of evolution. Now, within very short times, when erythromycin came out, within a year of it being used in Boston, about 20% of strains were resistant, and very quickly 50 to 60% of strains were resistant, and it wasn't able to be used anymore. But there are several urgent threats today. Clostridium difficile. A number of these are carbapenem resistant. These are brand new antibiotics, and we've got major resistance to these. There's many, many serious threats, drug-resistant tuberculosis. And there's some that are concerning threats, which are things like vancomycin-resistant Staphylococcus aureus. Why is that a potentially concerning threat? It's because vancomycin is the major drug that's used to treat MRSA, the multiple resistant Staphylococcus aureus. So how does this antibiotic resistance come about? Well, it may not surprise you to realize, since this is electron genetics, that it's genetic. And there's a number of ways that it can do it. The bacteria can just pick up the resistant genes that are just floating around in their milieu. they can actually have them injected into them by a virus that affects bacteria, called bacteriophages. They look like little moonlanders, don't they? And then in it goes, and then it gets incorporated. And of course, there's bacterial sex, where they conjugate, and they can pass on the genes to each other this way. And this is very handy for passing on big genes, whereas this is passing on little, tiny genes, which have a limited resistant. And the big resistant gene in Staphylococci is this MEK cassette, and it stops beta-lactam antibiotics. Now, beta-lactam antibiotics are things like penicillin that contain this ring. So how do bacteria overcome antibiotics? Well, they can do this in a number of different ways. They can inactivate the drug. They can use an enzyme to destroy the drug. They can modify the drug by adding a group to it that stops it working so it doesn't fit into its target site. They themselves can alter their target sites with a genetic allelic variation that prevents the drug working that previously worked very well. They can make another alternative protein that doesn't bind penicillin. So transpeptidase binds penicillin very well, stops the cell wall being formed, but it doesn't bind to the alternative, the penicillin binding protein, which binds penicillin very weakly. This can be used as an alternative transpeptidase to make the cell wall, therefore bypassing. You don't have to destroy the antibiotic. You can be really clever and evolve a different system that doesn't use the antibiotic, doesn't need the antibiotic to be there at all, doesn't care if the antibiotic is there because the antibiotic can't get at it. And then you can actually get rid of a metabolic pathway, amazingly enough. So sulfanilamide, which we know looks very much like PABA, and bacteria mainly need PABA to generate this pathway for the folate. There are some that just don't use PABA anymore, and they get this straight from the environment, like we do. We don't make this either, and we need to get it from bacteria in our guts. So methicillin-resistant Staphylococcus A, originally called meticillin, meticillin was changed to methicillin, and now the M stands for multi-resistant. Now, Staph aureus is a very interesting bug. 20% of people in this room will have it living on them where it causes no harm at all. It's a commensal, usually in the nose, but also in certain skin creases as well. It was found and discovered by Sir Alexander Ogston, and it was called Ogston's coccus, and he found it in pussy bandages from the surgical wards. Now, it has many, many virulence factors, this Staphylococcus. It can make a number of proteins, it can make a number of toxins that it releases that can cause things from food poisoning to abscess formation to septicemia, and when it gets into the body, it can be extraordinarily dangerous. It's also very tough and difficult to get rid of for a number of reasons. Not only just the resistance genes, but it tends to form slimes and lives in slimes. And here it is in real life on a slime. Antibiotics can't get into these slimes very easily. So these bugs are tough, they're hardy, and they're difficult to get rid of. And in fact, if they're living on heart valves, Staphylococci, for example, when they infect prostheses, it's sometimes you're in hospital for months with antibiotics trying to kill off the bacterial colonies. This is the beta-lactam ring. The very first resistance was an enzyme, beta-lactamase, that splits the ring. And this is busted penicillin. It ain't no good no more. So they invented methicillin, which has got another change in the shape here that doesn't fit into the beta-lactam ring. But it wasn't long before bacteria, particularly Staphylococci, became methicillin resistant. They worked out a mechanism. And the mechanism wasn't to develop another beta-lactamase. The mechanism was to develop that alternative protein, penicillin binding protein 2, that we mentioned that can carry on making the cell wall. And now we've got carbapenems, which have a completely different structure down here with these radicals coming off, which again makes it more difficult to be destroyed by the bacteria, and it's resistant to most beta-lactamases. But we have now bacteria resistant to this antibiotic. Now, what is happening with a lot of these, so there's different ways we can get the antibiotic-resistant genes passed around, as I've mentioned before, but what we want to look at here is this, the MEK-A cassette. This is the major. There are several variants of this MEC-A cassette, and they fall into two broad groups, really. Now, the one group is in hospitals, and hospitals had the original problem with MRSA. But emerging is the community MRSA. And what's interesting about the community hospital is that they tend to have smaller genomes for antibiotic resistance, but they've picked up a very nasty thing from a virus called pantine-valentine leukocidin, which is a very, very nasty toxin indeed that causes little pimples to turn into abscesses and abscesses to turn into cavities and to cause significant mutilation and risk of death, particularly amongst things like American footballers. Now, if you are carrying this, it's not going to do you any harm unless it gets into your body. But you can come into hospital and you can bring it into hospital with you. And if you do, it's going to have bacterial sex here, and hey, it's going to pick up the multi-resistant gene. And MEC-A doesn't just give you resistance to methicillin, there's a whole bunch of other antibiotics it's resistant to. What's more, they're not just having sex with each other, they're having sex with other species of bacteria and passing genes on. Now, Staphylococci and other cocci bacteria are rather resistant to just picking up genes in the milieu. They do need a mechanism to get it through. But they found that mechanism. So having a lot of multiple resistant antibiotic resistant bacteria in the environment is not a good thing. So who is hogging all the pills? Well, this is what antibiotics are used for in our society. They're used as growth promotion in animals. They're used as therapeutic prophylaxis in humans. You know, oh, have an antibiotic, it could get worse, take an antibiotic and go away. I'll come on to that at the moment. They're used in aquaculture. Loads of antibiotics are used here to make fish and prawns and various other shellfish that are grown in massive farms, particularly in the Far East, but also just off the coast of Scotland. They're used in culture for sterility and a number of other things such as towelettes. This is madness, absolute madness. The human race, I have come to a firm conclusion over the last 3 years, is stark raving lunatic and is bent on self-destructive path. Now, we keep on blaming the GPs for giving too many antibiotics. That's the general thing, because we like to blame GPs all the time now, don't we? On the new contract, they're not doing nights on call and everything. But in fact, they do a very good job. And it's darn difficult to get an antibiotic off a GP, even if you need one. But farms are using double the amount of antibiotics than humans. Now, you're not allowed to use them as growth promoters anymore. That's been banned in the EU. It's not banned in America, but it's banned in the EU. So what happens is Farmer Giles calls in the vet and says, I think my little porker down here has got the old snuffles. And he goes, yeah, that's not good. That could spread throughout the whole herd. We better treat all of them. And amazingly, this prophylactic use of antibiotic also works as a very nice growth promoter. And when you're talking about the difference between a farm going bust at $1 for a piece of meat and $1.10, then you need to get as much pork out of your animals as you can to keep afloat and keep in business. So it's not just the farmer's fault. This complete drive for cheaper and cheaper food production is part of this whole problem. We can't just look on this and point a finger and say, oh, you doctors are very bad. Oh, you vets are very bad. Oh, you farmers are very bad. We're all very bad because we go along to the GP with a bit of a snuffle and we expect to get an antibiotic. We've got to break out of this code. We've got to stop using them in aquaculture. In fact, it's amazing that 13,600 tons of antibiotics in 2009 were used on animals that were not sick. So which antibiotic would you like with your bacon, sir? And this is quite important, because you can phage type. We know where these come from. And in Holland, in 2004, where they have less of a problem with MRSA, they actually do routine screening, and to their amazement, a girl coming in for a routine operation was found to have MRSA. How come? Well, what had happened was she picked it up from the pigs in her farm. Not only that, this particular variety then spreads, and it spreads very, very quickly throughout the world. And it turns out that 24% of farmers were actually colonized with this strain. So is the meat. Now, it doesn't matter because it's not poisonous and you're going to cook it. But what is important is you're going to handle it before you cook it. And if you then do this, because you've got a little bit of an itch or you've got the onions are making you cry, you've now colonized your nose with MRSA, which may do you no harm. But if you go into hospital with it and you have a wound and you infect that wound with your MRSA, or you infect other people in the hospital with that MRSA, they have a serious, potentially lethal problem. Now, of course, this couldn't happen in England, could it? Well, Christmas, this Christmas, 2013, first time ever, MRSA drug-resistant staph found in Christmas turkeys in the United Kingdom. And this is really, really important. Just in November, this patient called BP was operated on in Vietnam, returns to New Zealand, and he dies of an infection that is totally resistant to everything. There was no antibiotic on earth could save his life, and he died. So I want you to imagine the future world where an infection is so dangerous that even minor symptoms can have you locked in confinement, such as Typhoid Mary, who ended up being locked up in secure hospitals until she died in New York, where you can't have routine surgery. There'll be no prosthetics, no hip replacements, difficult to put lens implants into eyes because the antibiotics are not going to be effective to stop them being infected. Unprotected sex, which isn't a wise idea anyway, will become seriously dangerous, like it did in 1915, 16 and 17. It's also important for the plants. Streptomycin resistance destroyed Michigan's orchards. It's now in upstate New York this year. If it destroys those orchards, no, we're not going to have no apples, no cider. I mean, this is going to be terrible, this world that we're imagining in the future. But there is something that can be done about this, and we do need to think about it. And it's thinking about this that enable people to go, do you know, if these bacteria are really good at switching just bits of the genome they want to other bacteria, is it not possible that we could make a gene and then use one of those mobile genetic element mechanisms and put it into a human cell? Perhaps put it into an embryo and let the embryo grow up, and an embryo that would otherwise be destined to form an adult who's going to die of, let's say, Huntington's chorea in their 20s or cystic fibrosis could live a healthy and normal life. Well, there are certain problems with that, and one of the problems is that actually doing germ cell, that's ovary, testes or blastomere, genetic engineering is a very, very dangerous thing because it's not just passed on to that person, it's passed on to every one of their generations. And if you get it wrong, and it's very easy to get this wrong because you don't know where the gene's going to be inserted for a start, you can end up with a number of problems. So the alternative approach is to go and actually try and transfect just certain cells outside the body and then put them back in. And then luckily for us, in the eye, we can actually treat the eye in situ. And you will know that last week some very, very exciting research data came out of Oxford from Professor McLaren's group, which I'll discuss at the end. So to transfer genes therapeutically, essentially we're going to do the same thing. Now, if we have an absent gene, there's a failure of function, which makes it easier because we can just replace this gene with a healthy gene. So we've got to identify the gene, we've got to make a copy of it, we've got to put it into a mechanism that can get it into the cell, and this could be a vector, it could be a naked DNA. Now, human cells are not very good at picking up naked DNA. We could package it into a virus, and viruses are very good at getting into cells. We could use a bacterial plasmid, which gives it some resistance, or we can use nanoparticles where we wrap it up in a lipid molecule. We get a very big payload of genes in there. But that's not the end of the story. Once we've got it in here, nucleic acids are in a hostile environment. There's lots of these things called restriction endonucleases that are present in the cell, and their job is to chew up nucleic acids. Their job is to prevent that cell dying from virus infections in the wild. And it can't tell the difference between an artificial nucleic acid and a viral nucleic acid. So it's doing its job, but it's a darn nuisance if you want to get this gene through here. But let's, for argument's sake, it does. It makes it through this hostile environment. It gets into the nucleus. It incorporates itself into the DNA, and it comes off, and then it's translated, and then that DNA goes off, and suddenly here we have the artificial gene transcribed as a new protein, which is the healthy protein. And that disease that is missing that gene is suddenly not a disease anymore. It is cured, as long as that is keeping on being expressed. Now, not all viral vectors allow integration. Some of the genome just is in the cell, and it decays very quickly, so you need to repeat the treatments several times. Even the ones that are integrated sometimes silence, and they need to be repeated as well. We'll get more problem if we've got a variant gene that makes an abnormal protein, and the abnormal protein causes the disease. It's a nasty protein instead of a nice protein. That's difficult, because if we just put in a nice protein, we've still got the nasty protein that's still being made, so we haven't dealt with that as a problem. But there is a technique coming out here called gene silencing. And we're able, with using small bits of RNA, to specifically bind, using zinc finger and a variety of other technologies such as CRISPR, into this dicer, which is a great big pair of scissors that comes up and chops up the DNA and breaks it down. Or for the other form of silencing, which is miRNAs, it can package it and store it, not necessarily destroy it. And this is the gene editing that we're using. We're using these enzymes. Here's the zinc molecule, here's the zinc fingers, and they're coming in, and very specifically, they're causing double-stranded breaks in DNA. Now, you can chop the DNA to chop out a bad gene. Say it was a mutagenesis you got from, I don't know, smoking cigarettes. You've got a gene defect there. We've got to identify what that gene is, chop it out, the DNA is then joined up with normal DNA again. The problem is with an inherited disorder, that doesn't work, because you're going to replace it with a bad gene again, unless you can do something about that gene. But what it's doing is allowing us to target very precisely, very accurately, certain bits of the DNA. Now, the problem with zinc fingers is they're a bit too small, so it's quite likely you can find some other bits that are not your gene of choice that you're aiming for, but some other random bits that happen to have the same sequence. And that can happen by chance, frequently enough in a genome that's full of billions of base pairs. So larger enzymes, such as the CRISPR and the iCRL homing endonucleases, there's less chance. This can only really be one place in the whole genome that's going to be like this. And this technology is advancing. So we can silence genes by chopping them out. We can actually home in, make cuts in the DNA to allow us to specifically insert a gene in specific places. And, in fact, this is a therapy that is going to be considered for Huntington's chorea, where these interfering RNAs are going to be introduced, and you can see the bad gene has been snipped out. And here we are. We're not going to make the bad protein anymore. This is the blastomere, which we mentioned before, and this is ex vivo, which is the more common way of considering, where we put the gene, we take the cells out of the body, we put the gene into them, we culture them, then we put them back into the body again to replace the missing part. And we can do this with non-viral vectors, and we can enhance the delivery of these, because just a non-viral vector just floating around doesn't get in, but we can use a gene gun with gold particles, magnetism. There's a bunch of different ways that this can be done, and we can design the polyplexes to be more efficient at being taken up by the cell. But most commonly, what I want to deal with in more detail are the viral vectors. So what we do is we load in the new gene into the virus, we sterilize the virus to make sure it's not going to kill you, and then we pop it into the body or into the cell culture, and here it comes, and it incorporates, and lo and behold, you've now got the gene here, and it can start making the good protein. Now, adenoviruses are not very good at doing this, and furthermore, adenoviruses cause upper respiratory tract and common cold problems. So if you're going to inject that into some person, you're very likely to end up with an immune response. And in fact, that did happen in one of the early gene therapy trials, and the young patient died. So adenoviruses, although they've got a big gene load and they're very common, are very difficult to use because of this immune response. But adeno-associated viruses don't cause disease so much and have a very limited immune response. So these are now one of the commoner forms of using gene therapy, particularly for eye diseases. Herpes simplex, you're going to hear a bit more about, because these can be used for neural diseases, but also they're very good for cancers such as brain tumours and melanomas, because you can put a suicide gene and into the vector and then home it into its target, and then it's going to kill cancer cells that otherwise couldn't be killed. So you're beginning to see this gene therapy has a little bit more going to it, and it's terribly exciting. The reason we haven't seen it widespread and used yet is it's terribly, terribly hard to get it right, get the right vector, make it safe, prove it's safe, get approval, and go forward. And then finally, retroviruses. Now, we all know what retroviruses are. AIDS is a retrovirus. So obviously using retroviruses, you've got to be very careful with them, and you've got to remove their infectivity to stop them giving you a rather nasty disease. Now, this, unfortunately, in 1999, was where young Gelsinger died in the gene therapy trial, just a few days after being injected from this massive immune response. And that was using the adenoviral vector. Now, using adeno-associated virus vectors, we can see there's a number of trials here in phase 1, phase 2, and one in phase 3 for treating cancer, haemophilia, Alzheimer's, arthritis, Parkinson's, cystic fibrosis. You know, hands up here who doesn't know somebody or has not come across one of those diseases on this list. But the one I've highlighted is this one, Leber's, which was the first successful trial, and that's what we're going to talk about when we come on to the eye section. Now, retroviral vectors, we've mentioned before, they came, and there was a, if you have this disease, which is the immunodeficiency, you're going to die of infections. It's very difficult to manage this disease because you're not making the gene that's necessary for immunity to work. And this is the boy in the bubble that you'll remember, David Vetter, who ended up with terrible psychological problems. He did eventually have a bone marrow transplant, but he died of Burkitt's lymphoma, transferred to him from his sister's bone donor, and he died aged 12. So, in fact, since there are problems with bone marrow donation, and to have a perfect match for bone marrow donation is extraordinarily difficult, gene therapy is going to be a really exciting way of transferring healthy genes and making healthy products for children with the various different genetic disorders that can cause this. And these lentiviral vectors now are used for Parkinson's disease, and we can see proof of concept where things are lighting up, it's going targeted to the right place, seems to be working, and there is some subjective evidence that it is helping. But what you want to know from me now is, yes, this is all theory, this is theory, but tell me about something that's really done, that's on the market. Well, here we are. Familial lipoprotein lipase deficiency. This is where you have a lot of fat in your blood. So this can kill you. It can cause pancreatitis and various other things. And there is a gene treatment for this, and it is the most expensive drug in the world. It's $1.6 million per treatment. Okay? Now, amaurosis, which we're coming on to now, is when the eye is not seeing, but it looks normal. And this was caused by a bunch of retinal and optic nerve diseases that weren't discovered until the ophthalmoscope was discovered. Of course, like all these things, an original invention by Charles Babbage, which we unfortunately gave it to a morphine surgeon who didn't do anything with it. So the Germans actually came on and invented their own and got on with the process of identifying these retinal diseases. You will remember that rods and cones, and they have little discs in them, and here's the disc, and then enlarging the disc, there's a lot of biochemistry going on in this. Furthermore, in the pigment cell that supports it, there's more biochemistry going on that can reconvert this form of vitamin A back into the form that can be reloaded into the photoreceptor. Defects in any of these genes can cause blindness. This is the one that is involved with Leber's. Others are involved with retinitis pigmentosa. And there's a bunch of different diseases that are associated with this, and they can be X-linked, they can be dominant, they can be autosomal recessive. Here's a good X-linked disease, colour blindness. And all New World male monkeys are colour blind. And the reason they're colour blind is that they've only got one X chromosome. And as we know, males are genetically deficient females. Now, you have the green receptor, for example, here. It's not really green, call it a mid-wave receptor. You've also got a blue receptor on your elsewhere, which are not X-linked. So you're a deuteranope. You've got two forms of colour receptors that peak. One of them in the mid-range here. For argument's sake, we'll call it green. Now, the female, of course, has two X chromosomes. So she is also a deuteranope, two receptors. But it turns out this allele is very polymorphic. That means there's lots of variance in the population, as I told you before, more than 1%. So some females can have a variant here that makes them with 3 receptors, which gives them the same sort of colour vision as we do as old world primates. New world primates split off and didn't evolve the gene duplication on the X that we have. We have this here on the X chromosome. So they did the gene therapy and they put in a human variant of this gene into the male monkey here, and lo and behold, they can start to see colours, the reds and so on, that they couldn't see before. And you think, how can you do a colour test on monkeys? Well, you can, and there's various things. He has to push that with his nose if he sees it, and if he does, you give him a drink of juice. I mean, it's that simple. It's like kids, isn't it? You know, they do something good, give them some juice in the bottle and hope grandma doesn't see you do it. And eventually you get the right sort of behaviour. It doesn't take long to potty train them if you really sort of put your mind to it. Now, so this could happen. Interestingly, one of those monkeys was called Dalton. And after Dalton, who worked out why he might be colour deficient. Incorrectly, as it turns out, but amazing thought that went into what he did. Now, Leber's congenital amaurosis, which also occurs in Briard dogs, which are a very old breed that we use for hunting, causes a wipeout of the... This is a dog retina, that's why it doesn't look like the other pictures. Now, Briards are particularly nice dogs, they're friendly dogs, you see them in films. Chewbacca in Star Wars was actually based on a Briard as well, for those of you who are Star Wars people in the audience, and there always seems to be one or two. Now, the gene that causes this is that RPE65 protein, the one that recycles vitamin A into a useful form. And if it doesn't recycle it, it builds up in the pigment epithelium and it kills the pigment epithelium. If the pigment epithelium dies, then everything above it starts to die, and eventually you end up blind. It takes a while to go completely blind, but you have very poor vision even in childhood. So what they did with 2 teams in the United States and one in the United Kingdom, they injected under the retina, pretty cool surgery this, the viruses that contain the proper version of RPE65. And lo and behold, they got improvement in vision for some of the patients. It was done as a proof of concept. And those of you who remember my very first lecture, when I showed the blind patient now after this treatment navigating through the maze without bumping into things. Now, it's not that they're going to suddenly read War and Peace by candlelight. It's not that sort of success. But what it is to slow the rate of blindness and to actually, in some patients, improve blindness, and also to show it's safe, which they did. Retinitis pigmentosa is another great disease to target because the commonest form of this is X-linked, i.e. a protein is missing. And it's boys, and they're going to come and recruit because they don't have that sort of danger signal that women do. So they're going to say, yeah, sure, inject me with whatever it takes. I want a bit of it. That's the normal fundus, and this is the fundus of retinitis pigmentosa. So there are a number of treatments, gene treatments, that may alter the progress of this disease. And then finally, I want to mention choroideremia, which is where the big trial has just come out of Oxford, again showing positive effects in this otherwise relentlessly progressive disease. Now, can we use this for other treatments? Yes, we can. We can use it for things like macular degeneration. Instead of having an injection every month now, we can give you a viral transfer, potentially, and that makes the anti-VEGF for you. There's some problems with that. God made VEGF for a reason. Now, I know when it goes wrong, you get macular degeneration, but it does other things as well. And we don't really fully understand this to permanently give someone an anti-VEGF virus that could spread out of the system, go somewhere else, cause some terrible problem. So, but anyway, these trials are underway, and the good thing about the eye, though, as opposed to other organs, we can limit it to the eye, so it's less likely to spread out and cause damage elsewhere. I just very finally wanted to just keep you alert to some emerging treatments. Optogenetics is one. If you have no retinal receptors left, such as in retinitis pigmentosa, you can infect with virus an artificial light receptor, which means the cells above can now react to light and get the pathway working again. We can use stem cells. We've got artificial vision. I've just come back from the artificial vision conference in Aachen, where various different types were discussed, both the American Argus, which is this, which has restored some useful vision in patients, and one patient can actually draw letters from complete blindness. I mean, this is miraculous. And then finally, you can get an inkjet to make a retina. And in Cambridge, Keith Martin, who's the professor there, he got a piezoelectric printer, put some cells into it, and was able to print out a retinal mosaic with ganglion cells and some structural cells for it. Well, this is extraordinary. So, ladies and gentlemen, I hope that you found that interesting and stimulating, and that will help you understand some of the newspaper articles that you're going to see, or you're going to be bombarded with over the next few years. I'd like to thank my patients, as always, and the colleagues and staff at the hospital. Without them, I couldn't have done any of this work. Of course, the Gresham College staff and the Museum of London AV, which was fantastic today. Thank you very much indeed. And if you have any burning questions or things, you can contact through the college. And finally, a picture of corneal stem cell, being a corneal person with an amniotic membrane overlying it, in a young Indian girl who had complete grade 4 burns from lime splash into her eyes, which is a very common paediatric emergency over there. Thank you.